Okay, everyone. It's time to start. So, first of all, thank everyone for being here. And it's my great honor to invite Klara for today's seminar. Dr. Scharnagl is the fungal ecologist from UC Berkeley, and she serves as Tucker Curator of Lichenology at the University Jepson, Herbaria. She got her PhD from Michigan University and finished her postdoc in the Turbo lab. Now she's investigating the evolution and ecology of tidal lichens. So now let's warmly welcome Dr. Scharnagl for today's talk.
Oh, perfect. Yeah, you can see all the pretty pictures better. Thank you so much for the invitation. I have had a spectacular day so far, getting to converse with a number of people in the department and get inspired by ideas. And there's probably a number of you I'll be following up about potential collaborative projects with, because that's the nature of these things. And the nature of lichens also. So yeah, oh, perfect. All right. I'm going to begin with this quote by Vincent Zonta. Lichens are familiar to everyone, known to no one. And you'll kind of see this throughout today's talk. And basically, every single one of you has been in the presence of lichens, probably just a matter of minutes ago, if not at least sometime today. And you've probably seen lichens all over the place, whether or not you actually recognized them when you're seeing them. And this actually not only applies to your experience with lichens, but to our history of our experience with lichens, which is what I'm going to lead you through today. And in fact, normally I begin talks by saying, oh, wait a minute, what actually is a lichen? But since that's the whole crux of my talk, we'll get there. Or we won't. We'll see by the end of the talk. Yeah, so without further ado, here's sort of the structure we'll be following. So I'm going to begin with our history of perception of lichens. And really throughout this entire talk, I want to emphasize the importance of how our perception and language influences our understanding of the world around us. And that's going to be a theme that I keep circling back to throughout our exploration of what actually is the lichen symbiosis and what do we know about it. So from the history, we'll then come to the classic definition that was held for a long time, and it is actually still the way that lichens are most often introduced. But then, of course, I'll introduce you to some recent discoveries and ecology that has led us to some newer definitions. And then finally, hopefully give myself plenty of time at the end to discuss some conceptual challenges and biases that might still be in our way to a full understanding of the nature of the lichen symbiosis. Also, just want a quick check. Can anyone at the back hear me? I can project more. Okay, great. All right, so we'll begin with the history. So lichens actually were originally thought to just be excretions of the rocks or bark upon which they were found. So in this picture, it would have been assumed that none of this was actually, living material. All of it was just stuff being effused from the rock. So that was our original concept of lichens. And when I was talking about this talk with my mom, she said, well, yeah, I mean, that was my perception of lichens to start with. I saw these colorful things on bark and on rocks, and I just thought it was part of the bark or the rock. And so many of us have come to lichens from this very early perception of it. And this actually led me to think about a much more recent quote about lichens by tropical ecologist Dan Jansen. Ecologically speaking, lichens are hardly more than intricate scum on the surfaces of rocks and trees that are too harsh for angiosperms. So of course, in this text, he was very focused on the angiosperms, was really making a case for them, but that relegated lichens to the role of intricate scum. So once it was finally recognized that lichens were actually alive in some sense, they were lumped with plants, but of course considered very basal. And in fact, so were most of the fungi. Fungi were lumped with plants also. And they were also considered basal because they weren't doing cool things like photosynthesizing and becoming trees. So when lichens were studied at all, they were studied by people who happened to be looking at plants. And of course, I couldn't help but include Ernst Haeckel's lichen page from his art forms of nature, because it's just really fantastical, surprisingly accurate as well. So yes, so like I said, consider basil, and if you wanted any proof of that, Linnaeus described lichens as the poor trash of vegetation, which is terribly unfair. But all right, Linnaeus, that's all right. You did something. So before we get deeper into lichens, I wanted to take a moment to say that we as humans were using lichens long before we actually understood what they were on any level. And so there's evidence, written records from China from the 1500s of use of lichens in medicine. Many indigenous groups, of course, were also using lichens medicinally. When I went to visit Heidelberg, Alaska, the elders there said yes, for many generations, of course, we've been using usnia and other lichens medicinally. Lichens have also been used as food, only certain lichens, and I always introduce this with a caveat. Please don't go out and start eating lichens, because many of them produce secondary metabolites that at best will give you a stomachache and at worst could do something much worse. So when in doubt, just don't. Lichens have also been used as dyes. They were actually some of the original sources of dyes, particularly for complex colors or hard to get colors like yellows and purples. And actually still lichens are used as dyes, more on the level of hobby these days, but still very much used. And then as perfumes and fixatives in perfumes, so both as a source of scent and as a way of fixing that scent. And I actually have an image of Pseudovernia furfuracea here, because this was found inside of the ancient Egyptian mummies. So there was already an understanding that these lichens were providing a fragrance that helped the mummies not smell like dead bodies. And the coolest fact about that to me is that this lichen is not known to grow in that region. And so not only did the ancient Egyptians have knowledge of what this lichen could do, but they had knowledge of it from somewhere else and were in fact importing it for this specific purpose. So super cool. So again, we've been using lichens for a very long time. So finally, the more people looked at lichens, the more they were like, what's with these like green round cells in here? What's going on with that? And they don't look like the tangled fungal hyphae that are around the green cells. And finally, some folks, including Schwendener, proposed this dual nature or dual hypothesis of lichens. These aren't one thing, they're two. And the term symbiosis is found in a few places, but it seems like it was popularized to describe the lichen association. And I will say, when Schwendener and others proposed this, of course, they got tremendous backlash. Total hogwash. What is this dual organism nonsense that you're proposing? But eventually it caught on. So just to define this, symbiosis has many definitions, but I would say it's the long-term and intimate association. The living together is literally the translation of two or more organisms. And so this has nothing to do with whether that association is positive or negative or somewhere in the middle. Symbiosis encompasses all of those kinds of variants. So that's the history. We've come from not even recognizing these things as living to actually recognizing, wait, these things are alive and there's more than one thing involved there, which brings us to our classic definition. So most introductions to lichens these days, most textbooks will give you this: fungus plus algae equals lichen. So simple, beautiful. It's like even written out as a mathematical equation. This may be oversimplified, but basically, even I myself give this definition often when I'm introducing lichens. So lichens are a symbiotic association between a filamentous fungal partner and a photosynthetic partner, which can be green algae or cyanobacteria. or sometimes both. And so then, of course, the question arises, why? Why this dual nature? Why are fungi and algae living in symbiosis? And there are just as many answers to that question as there are definitions of lichens, but I'll go into it a little bit. So we'll begin. This is a microscopic image. This is a cross section through a lichen, as though you're cutting a slice through a sandwich. And so, ooh, yeah, okay. So most of what you're looking at is fungal. So up here we have thickened hyphal cells. This forms the upper cortex. All of these rounded structures are our algal cells. So that is the photobiont layer. So it's very close to the surface, but not exactly on the surface, so that it has that protective layer over top, but it can still get all the sunlight it needs for photosynthesis. Beneath that is this more thinner fungal hyphae, a lot of air pockets. This is the medullary layer, where a lot of that sort of gas exchange takes place. And then some, but not all lichens, also have a lower cortex as well. What I love about this is, here's the same cross-section of a lichen next to a cross-section of your leaf, where we have our upper protective layer, our high concentration of chloroplast layer, our more open gas exchange layer, and our lower protective layer. And so lichens have developed this, potentially optimal arrangement for promoting the photosynthesis of their algal partners. So the idea again here is they're optimizing that photosynthesis because the algae are photosynthesizing and giving some of those photosynthates either in the form of sugars or sugar alcohols, possibly some lipids, although we're still studying that, to the fungus. And in exchange, the fungus is capturing water and mineral nutrients from the environment and also offering that sort of protection to these algae. So within the lichen, the algae are far more protected from herbivory and pathogens than they would be if they were free growing. So even just that description of what's going on in exchange between these two partners has led to a lot of variations on that classic definition. So back in 1913, Fink said, a lichen is a fungus which lives during all or part of its life in parasitic relation with an algal host and also sustains a relation with an organic or inorganic substratum. By that, he was describing the fact that these can be found growing on living things like trees, but also on inorganic things like rocks. But yeah, so parasitic relationship. This is another stab at it. So lichens are a biological and not systematic group. So again, acknowledging that there's multiple things involved here, and possibly even within the fungal and algal groups, multiple different groups involved. Each is basically a stable, self-supporting association. So we already have the sense that maybe lichens are some sort of nice closed system arising from the symbiosis between a fungus, what we call the mycobiont, and an alga or cyanobacteria, the photobiont. So the way I described what the fungus provides to the algae has led to this sort of definition. So Trevor Goward commented that lichens are often regarded as fungi that have discovered agriculture. And if you think about it, it kind of makes a lot of sense. I mean, what do we provide to our food crops is water, nutrients, protection. Basically, the fungi are doing a similar thing to the algae. And so this has really stuck as a concept, although I think, again, there's probably more nuance than that. I like this definition by Rosemary Honegger in 1991. Lichens are the symbiotic phenotype of nutritionally specialized fungi that live as ecologically obligate sorry, there's so much jargon in here, but it's still nice. Ecologically obligate biotrophs in symbiosis with algal and or cyanobacterial photobionts. So again, she's really trying her best to capture everything that's going on here. And I will take this moment to say that yes, as far as we know, the fungi are actually obligate in this association. We don't find them free living outside of the lichenized state, whereas the algae can often be found free living as well as in the lichenized state. So it's not a completely balanced relationship in terms of who is obligate or who needs that symbiosis to survive. Which again, some people have used to make more of a case for that fungi as farmers. But yes, I really wanted to emphasize this idea of the symbiotic phenotype, because when we isolate the fungus, like I said, we haven't found it free growing. But when we isolate it in culture in the lab, it never looks like the lichens that it was isolated originally from. And basically, all component parts of the lichen are needed to ever form what we recognize as the lichen. So that was the classic definition, which, as you can see, is already starting to get more complex as we try to understand the nature of this relationship. So let's add some more complexity. So the symbionts are not always the same. Before we had molecular tools and ways to sequence DNA, these were considered two different species. And of course they are. They're morphologically super different. Even their reproductive structures are different on these two different things. But in fact, both of these arise from the fungus Sticta canariensis. But this one is in association with green algae, what the authors are calling the chloromorph. And this is in association with cyanobacteria, what they call the cyanomorph. So just associating with a different partner can lead to an entirely different symbiotic phenotype. And there's multiple examples of this. It's not that every single lichenized fungus can do this, but again, there are multiple examples of this happening. And sometimes the difference is even more radical than this in terms of what they look like. So with molecular tools, we've been able to uncover both cryptic species as well as things that we thought were different that are actually the same. And again, when I'm talking about species, I'm talking about that main fungal partner in this. So more complexity already. And then, so it's not just that you can associate with different photobionts, but there may actually be multiple fungi involved. So lichens splashed into the popular media in 2016, when they also splashed onto the cover of Science, when it was discovered that in certain macrolichens, and both of these are examples of macrolichens, that basically just means lichens that are three-dimensional. Microlichens refers to all the crusty things that could be mistaken for just excretions of the rock or bark. So yeah, so in certain macrolichens, it was discovered that under that upper cortex layer were these basidiomycete yeasts. And once they were found, the lichenologists started looking in more lichens, and they were found again. And so it all got really exciting. And it's like, oh, wait, there's multiple fungal partners involved. And this, like I said, this got really popularized across both the science world and popular media. And I am using this as an example, but this is just one of many articles that came out around that time. But I wanted to use this as an example of a caution in sensationalizing biology. So basically, as soon as this was out, and Ed Yong's tagline really sums it up nicely, biology textbooks tell us that lichens are alliances between two organisms, a fungus and an alga. They are wrong. And I feel like that's a bit of a blanket statement. It's as much of a blanket statement to say that all lichens contain this extra partner, because in fact, we do not know that to be true. Our primers are getting better and better, so we are finding more and more of them, but certainly in those microlichens, like I was mentioning, we don't tend to find them as often as in the macrolichens. So that still means that they're probably playing a really cool role in the macrolichen symbiosis, but it doesn't necessarily mean that they are a part of the definitive definition of what it is to be a lichen. So that's the caution tale that I just wanted to share there, even though it's a really cool discovery. So in the meantime, again, with all of our cool molecular tools, we've realized two important things. First of all, that lichenization has evolved multiple independent times, both in the fungal lineages as well as in the algal lineages. And on that note, the algae are far more diverse than we thought previously. So when we were just identifying these algae based on morphology, they basically, either looked like little filaments or little green spheres inside of the lichens. And so we're like, okay, there's a handful of species and also some cyanobacteria. And now that we have molecular tools, we're realizing they're much more diverse. Bonus, they're much more diverse in a single lichen. So usually a lichen will have, again, that sort of main fungus and main algae, but then smaller populations of other algae in there. And there's only been a few studies on this, but they've shown, and actually the same kind of study has been shown in corals, which also have photosynthetic symbionts, that those photobionts in low density, when environmental conditions change, might then become the dominant photobiont because they're better adapted to that particular situation. So it seems beneficial to have a bunch of potential partners around to deal with different kinds of environmental conditions and stressors. But again, that particular work is very much in its infancy, and we need a lot more data to really know what's going on there. But suffice to say, there's a lot of algae in lichens, not just across different lichens, but in a single lichen. So the takeaway from this section is lichens are rarely, if ever, just a one-to-one symbiosis. And of course, they aren't just a one-to-one-to-one symbiosis either. So that leads us, okay, what do we do with all of this new information? It's time to go back to the drawing board and get some new definitions set up for lichens. So with all of that recent level of discoveries, what do we know? Oh, yes, so heading towards a new definition of lichen symbiosis. All right, so again, just like Rosemary Honegger's definition, Foxworth and Grube are really trying to encompass absolutely everything of the updated version. So I'll read it out. A lichen is a self-sustaining ecosystem formed by the interaction of an ex-habitant fungus and an extracellular arrangement of one or more photosynthetic partners and an indeterminate number of other microscopic organisms. Whew, oh my goodness. Yes, but also, holy smokes, that's way more complicated than our nice little mathematical equation we had a few slides ago. But this is our new understanding of lichens and the lichen symbiosis. If it's hard to look at a big chunk of text, I'll show you this fabulous figure. I highly recommend this paper for anyone who's really interested in the latest on the lichen symbiosis. This is Allen and Lindemer. I'm pretty sure the title is A Call to Reconceptualize the Lichen Symbiosis. And they have fabulous figures. I actually stole like 2 of them for this talk. But yeah, they also go into detail exploring what we know. So here we have a lichen. You see it, you recognize it as a lichen, this particular lichen. And it has, of course, our classic components. It has that main fungal, filamentous fungal partner. It has probably a main photosynthetic partner, but also probably subpopulations of others. It has a whole mycobiome or community of endolichenic fungi. And these can be forming any sorts of associations, everything from commensals to possibly involved in the symbiosis itself to actually parasitic on either the lichenized fungus or the lichenized algae or both. Meaning these guys up here. There is, of course, a whole bacterial microbiome, and a lot of that has been demonstrated using metatranscriptomics and proteomics to contribute to the functioning of the lichen symbiosis, whereas others might be commensal or even pathogenic themselves. There's quite a diversity of microinvertebrates in a lot of lichens, in and on a lot of lichens. And they are absolutely playing a role in this miniature ecosystem here. And then they added secondary metabolites as sort of the second or an additional category. A lot of these are understood to be made by this main fungal partner, but of course, a lot of these other organisms are probably contributing to that, either contributing components to the pathway or contributing their own secondary metabolites to this. And these are known to play an important role in maintaining that symbiosis. either in forming sort of protections in terms of antimicrobials or even sunscreening compounds, but also possibly involved in signaling and other sorts of things. So that's why they included it as like its own sort of separate component there. So I like this quote by Lynn Margulies. Lichens are examples of innovation arising from partnership. The whole is far greater than the sum of its parts. And that's absolutely true. So take any of these components on its own, and it does not a lichen make. But bring them all together, and it does make a lichen. And a lichen that seems fairly stable and self-supporting. So yeah, I like the way that she summed that up. And again, it also refers back to that symbiotic phenotype. So take any of these individual parts, and none of them will form the lichen the lichen symbiosis, but they also won't form this structure. So we've gone on a whirlwind journey so far from the history of lichens and our understanding of them to arriving at this classic definition that was beautiful. I mean, for a long time, lichens have been described as the poster child of symbiosis. The term was made for them. And it's this great, beautiful, simple equation of fungus plus algae. And then these recent discoveries that have led us to these more complex and yet in a way more encompassing definitions that, and I'll get to this in the next slide as well, that challenge the way we talk about organisms, individuals, life, symbioses. Because there's complexity there, and yet these are entities that can be recognized and discussed as themselves, as lichens. So before I get into some of the challenges, I just want to give some more facts about lichens. So, and yet there are biases in this too, which we will explore. So lichens are known as polyextremophiles. This means that they can survive not only in some kinds of extremes, but other kinds of extremes as well. So is it super dry? Is it freezing? Is it super hot? All of these things, there are lichens that can survive those conditions. Now, just like the Basidiomycetes, this has led some popularization of lichens to lead people to the conclusion that all lichens are polyextremophiles. All lichens can survive all conditions, which of course is totally not true. Different kinds of lichens are adapted to different kinds of conditions, and some lichens are in fact super duper sensitive. So lichens in the tropics can't just be plucked from the tropics and thrown into the Arctic tundra and be like, go for it, you can survive freezing, right? And they're like, no. Yeah, there's different adaptations to different environments, and there's even specificity of what they grow on. So not just the conditions they grow in, but what they grow on. In general, and of course there are a few exceptions to this, lichens that grow on rocks are not the same as the lichens that grow on trees, which are not the same as the lichens that grow on leaves, which yes, there are lichens that grow on leaves, which again are not the same as the lichens that grow on soil. When we get to lichens that grow on concrete and plastic, I don't know what to tell you. Often they originate as lichens that grow on rocks, but not always. So yes, polyextremophiles. This is actually an example of an endolithic lichen in the dry valleys of Antarctica, which I was talking with some folks about earlier today. So you can see that algal layer actually inside the rock. So pretty cool. So yes, again, some of them can deal with extremes. Does that mean that they can travel to outer space? Yes, it does. So if you've been following the recent news, there was just lichens splashing once again. I'm sure far and wide, everyone saw this, that they traveled to outer space. This is not the first time that lichens have been taken purposefully to the International Space Station, put on the outside of it, and exposed to conditions of space. But it's the longest period of time that they've done so. So this was 18 months. The first experiment was just like a day or a few days. And then they've progressively increased the amount of time of exposure. So there were two experiments done with this most recent study. One was they had certain sort of like filters in place. So the lichens were on the outside of the space station, but just exposed to Mars-like conditions. And then others were exposed to everything, the vacuum of space, the crazy temperatures, the gamma and like ultraviolet radiation, all of it. And they were brought back to Earth and assessed and rehydrated. And the ones in the Mars-like condition had, I think, between 60 to 80% survival rate in terms of their cells. The ones exposed to all of space had 35% survival rate, which sounds low, except they were just exposed to outer space. That's actually really high. So lichens are really cool. And this is an example of another one that was used in earlier experiments, but I love it because, you know, it kind of looks like some of our popular ideas of what an extraterrestrial might look like. So it's fabulous. This is actually occasionally a vagrant lichen, which means it's not attached to anything. It kind of rolls around like a tumbleweed, but adapted to pretty extreme conditions. So yeah, so like I said, some lichens can deal with really intense extremes. Again, pluck a lichen from an old growth forest or from the tropics, and they will not survive this. So given all of that, you would think that growing lichens in the lab or in a growth chamber would be easy. They can survive outer space. Of course they can grow in my growth chamber. No, is the answer to that. They do not. They grow very slowly, if they grow at all. And the problem with growing slowly is if you have any contamination in here, it can quickly overtake the culture you're actually trying to grow, and you don't end up with your lichen. So both in terms of actually growing whole lichens in growth chambers, as well as culturing those two main components, that filamentous fungus and the algae, we run into a lot of difficulties. Again, not impossible, and our techniques keep improving as we understand these better. But isn't this beautiful? It's a blob of fungi and algae that have been brought together on a plate. This, you might notice, doesn't look anything like this. And yet these parts came from that. So clearly, when we're trying to recreate or resynthesize lichens in the lab, we're missing something. So whether it might be some members of that ecosystem that I introduced you to a few slides ago, or whether it's some sort of environmental conditions that we haven't gotten just right, all we know is that lichens can survive in space, but we still can't recreate them in the lab. And as someone who studies lichens, I kind of love that. I love that they resist us trying to control them. And yet it can be frustrating when we're trying to do experiments to understand what kind of genes are expressed when these partners come together. So pros and cons, but defiance little pieces there. So not only is growing lichens difficult, but also a lot remains unknown about how lichens form. So there's some basics. And again, if you want a nice paper to read about this one, which I believe is called How to Build a Lichen, also has really great diagrams and is just a really great paper to read. So they're showing here basically the different steps. This is an algal cell. This is fungal hyphae. So there's like some signaling going on in advance. They make contact. The fungus starts growing around the algae. Exchange starts taking place. And yes, I think it says it on there. Yeah, increase in algal cell size. In general, when we look at the algae inside of lichens, they are much more spherical and much larger than they are in their free growing or in culture conditions. So there's something about being in the lichenized state. And again, you might be like, it's just like farmers, we're trying to maximize our yield. But there's probably a lot more going on there that we don't know. So a lot of this understanding comes from lab studies, but some cool studies have been done. Again, this was by putting plastic cover slips out into nature, but they were out in nature, and so they were able to collect these at different time points. This is a fungal spore germinating and interacting with algal cells nearby and starting to create basically a clump. I think they did take this all the way through to actually seeing it form a recognizable lichen. So again, that's different from what we're able to do in the lab. And clearly there were some environmental components added to that were needed. But a lot about this, and I would say particularly about this stage, is very poorly known. And a lot of that is because we can't just go out into nature and observe this taking place. This is happening at the microscopic scale. So that remains a challenge in our understanding. And it seems like kind of a crazy challenge, given that lichens are terrestrially ubiquitous. There's so many of them. They're forming all the time. How is it happening? So anyway, so we still don't fully know. So that's another challenge. So I want to come back to what I mentioned at the beginning, which is the role of perception and language in our understanding of life, but in this specific example of the lichen symbiosis. So in terms of some of those earlier perceptions of lichens, how inclined are we to study excretions or scum or trash or basal things? This language has impact, and this perception has impact. And even if, okay, we study them, how inclined are we to think that they are important or important enough to be protected? So this is something that even now that we have this better understanding of lichens, I still run into as a challenge. Why study lichens? What even is the point? So this is something that is very important. On that note, it also influences how we understand the nature of that relationship. So a lot of these terms are terms I used in this talk or alluded to, or are terms that are used to describe the lichen relationship. So I'll read them out again. Parasites, farmers, hosts, symbionts, partners, ex-habitants, inhabitants, mutualists, obligate, self-sustaining, capture, individual, body, cooperation. And this is just to list a few. So, I would imagine each of these words not only has a very different kind of meaning, but probably elicited a very different emotional response from each of you. And so again, that not only influences how we study and understand the nature of these things, but again, it also influences how we can encourage others to appreciate them as well. Oh yes, so I wanted to just quickly explore, based on some of those terms that I just listed, I've been thinking a lot more about this lately because not only are lichens in the popular literature a lot, but so are mycorrhizal fungi and their relationships. And so in symbiosis, who is the host and why does it matter? So we'll start with the human microbiome. So this is an interesting figure that I came across, which basically says the complete human is not complete until we add the invisible microbiome, but that's still thinking of it from, what makes a human, which could be a very important way of thinking about it. But I would love to think, and again, if you want to read more on this, Lynn Margulies did a lot of work sort of thinking from the microbial perspective. So are we humans hosting a microbiome, or are we the manifestation of the microbiome's world, so to speak? I'm not putting it very eloquently, but basically she tried to make that argument. And again, with lichens, we've actually gone back and forth as to, and it was even in some of the quotes, as to describing who's the host. Is the fungus a parasite to the algal host, or is the algae, the little crop of the farmer fungus? And so again, just defining who's who really influences our way that we approach studying these relationships. And that absolutely goes for plants and mycorrhizal fungi, which are almost universally studied from the plant perspective. And I think we really need to start thinking about it from the fungal perspective. And maybe we'll come to the exact same conclusions, or maybe we won't. And I think it's worth doing. So yes, so coming full circle, and again, this is a fabulous figure from that Allen and Lindenberg 2022 paper, where they really get into the idea that lichens are not just one thing. Lichens are not just two things. Lichens are whole ecosystems. So each lichen you look at is a miniature ecosystem, and with all the component parts and all the ways that we can think about things. So, it has one or more sort of dominant fungi one or more of those photobionts, and then a whole myco and microinvertebrate biome involved in that. And then you can start thinking about each of these as a population of ecosystems. So here they're suggesting that the species of that dominant fungus are the same, or a whole community, where we're starting to look at lichenized fungal species, but even within that, different genotypes, and start approaching these more from that ecosystem community perspective when we're asking our questions. And again, maybe we'll come to the exact same conclusions, or maybe we won't. And of course, there's the added layer of lichens as ecosystems in ecosystems. And again, I think we as lichenologists are really excited about this, but we're still finding the right language to really talk about this well. But basically, you have all this embedded within a larger ecosystem, aka an old growth forest, or the Arctic tundra, or even smaller than that, a tree, a boulder, and what are their interactions there, as well as their interactions here. So we have this sort of nested ecology, nested symbioses going on. And again, we're all very excited about this, but we're still figuring out the best way of talking about it. So I really love this quote by Merlin Sheldrake in his book Entangled Life. Lichens, in some sense, are microplanets, worlds writ small. And so yeah, so here we have a whole bunch of those crust or microlichens, and each of these is a little world. And again, I really love thinking about it that way, even though it's complex and challenging. And I have 2 more slides. I think I'm doing good on time here. Plenty of time for questions afterwards. So this leads me my next slide, which I would be remiss, given the most recent stuff that's been splashed across the news, to not mention, which is the importance of collections. That's natural history collections, specimens, understanding the lichen symbiosis, and I would say all of life. So again, this is a little world. It's collected at a very specific time, in a very specific place. There is no other little world exactly like this. So when it's put into a collection, it is a timestamp of which there is no exact duplicate. And so we can gain so much from these little worlds and preserving them. And if you think about it, collections were started long before a lot of the tools that we now have. So when early naturalists were starting to collect things and actually think about preserving them long term, they didn't have DNA sequencing. They didn't have stable isotopes. They didn't have scanning electron microscopy. All of these are tools that we've used more recently to gain so much more insight from collections, as well as from current specimens. And so a lot of arguments have been made to me as a curator. You have all these digitizing efforts going on. You're taking images of your specimens, you're entering all the data online. So we have that spatial data, we have that time point data, we have the image. A lot of collections, including my own, are starting to DNA barcode our specimens. You've got everything you need. Why hold on to the specimens? Well, I think it's that argument. We don't know what technologies we'll have 100 years from now that we'll be able to apply to these specimens to gain even deeper understanding, and not just deeper understanding of like life in real time, but life historically, which gives us so much more profound insight on the nature of life and the nature of our relationship with it. So yeah, preserving these worlds for current and future study, I think, is not only useful, I think it's essential. So with that, I just have some closing remarks. So again, when I study lichens, they're often framed to me as sort of the exception to the rule. It's like, okay, you know, these people are studying monkeys, these people are studying oak trees, but oh, you're studying lichens. They're a symbiotic association, woo! When in fact, all of life is symbiotic. So to me, the usefulness in studying lichens is not that they're the exception, it's that they're the rule, that there are these nice, compact little systems for asking questions about those symbiotic relationships, which we then can apply to all other symbiotic organisms, ourselves included. So not only is all of life symbiotic, but the nature of that symbiosis exists along a constantly shifting continuum. You know, as I've shown you in the history of our understanding of lichens, we've gone back and forth, parasites, mutualists, you know, people even use like terms like altruistic or, you know, other things. And no, it's not any of those things. It's a relationship that retains stability while oscillating along that continuum for the growth and survival of all of the components involved. And I did not come up with this hashtag, but I really love it. And I feel like it sums up this slide very well. We are all lichens. And with that, I will take any questions.
That was really great history of studying lichens. It's interesting, like plants oozed out of water and had an endosymbiont that could do photosynthesis between trees. Why has the fungus, which needs its or cyanobacteria to survive, just as a symbiont, not captured and made it an endosymbiont like a chloroplast? And was it that the plants were already doing it, and so there wasn't the niche? Or
This is such a great question because, yeah, lichenization has evolved multiple times. It seems evolutionarily like a good idea to do so. And so, yeah, why wouldn't it go to that next level of just, you know, if this partnership's so great, let's make it totally permanent. And I don't think we really know the answer to that question. Interestingly, there's the whole thing that I didn't get into at all of how these things propagate and disperse. And so a lot of the fungi sexually reproduce so that the spore has to go off and land somewhere. And likewise, when the algae are free growing, they're sexually reproducing, and also they can be free growing or in the lichen. And so I think there might not have been a pressure there per se. It still is a fascinating question. And again, I still don't think we fully know the answer. I think there's one or two examples of fungi that actually do have algae inside. They're very little studied, although I think one of them, the genome either is in the process of being sequenced or was just sequenced within the past couple of years. And so we're about to have some really cool data to play with to try to get at that.
so there are some.
There are like one or two examples that we know of. Yeah. Yes.
I have two questions.
Mhm.
One question concerns how you grow the lichens in a plant. Like, do you deplete the nutrients so that the fungus is forced to undergo symbiosis?
Yeah, so we used a few different approaches because the examples that I showed there were from an experiment that I ran during my postdoc. And I first wanted to generate a lot of biomass of the fungus and the algae separately before then bringing them back together. And to do that, I actually used sort of a regular medium, which had stuff that the fungus wanted in there to bulk up that biomass. But then when we put them together, yes, we used a totally nutrient depleted medium for that. Yeah.
One last question is, you described the lichen as a mini ecosystem, but when you think about it, like we ourselves are like mini ecosystem.
Of course.
Like plants, the different cells of a plant, some photosynthesize and some develop nutrients, similar to how the fungus and the algae play a role in lichens. So would it be okay to view lichens as also a bona fide individual, like a plant or a human?
Yes, yes. But I think that is the language that we're struggling with, because I absolutely think we can call each lichen an individual in that sense. But then I think people struggle with that because we think of individual as both singular and indivisible, which of course is not really true, but maybe that's not true of anything that we refer to as an individual. So yeah, it's playing with that language, and maybe if that understanding itself will evolve along with our understanding of all of these systems. Yeah, I feel like I saw it from the middle somewhere.
Yeah. Jerome.
Yeah.
You raise your hands.
I love the actual working on vaccine, the allergy fungal interaction.
Uh-huh.
So I actually thinking quite interesting that some lichens are actually parasitic lichens. How do you explain that?
Oh, yes. So there are lichens that we refer to as lichenicolous lichens. That is, they grow on and even parasitize other lichens. And to me, that is just an example of worlds upon worlds. You know, one of my favorite
But
talks.
that would
Oh,
affect
yeah.
the interpretation. Can you have both parts? You could do photosynthesis, but they actually parasitic.
Yes, that could influence things. And depending on which parasitic lichen we're talking about, they do different things when they're parasitizing the other lichen. Sometimes they do take over its algae. Sometimes they don't. And so, yeah, to me, again, it's just sort of sliding along that same scale because a lot of these just seem to have landed and started growing and becoming a lichen on top of another lichen, but they have this additional mechanism in place to just fully take over that other lichen basically as a substrate, but also as a food source. So I agree with you. I think there's a lot more to explore in how we think about the nature of that relationship. Yeah, come back to your question now.
How do microlichens attach to rocks?
Yes. There's a number of mechanisms that they use. They can either just sort of use hyphal force to get a foothold or a hyphal hold into their substrate. Sometimes along with that, especially with rocks, they'll secrete minute amounts of acid as well in order to dig in. Yeah. Yes.
So I think you mentioned that when you try to recreate association in the lab, they definitely grow different than in nature. So how hard or how difficult is to extract DNA or RNA from the like how they grow in nature, just like the structure, and then compare that to like your grown in culture and like I don't know, do RNA-seq analysis or just compare what kind of genes are expressed? What is different?
Yeah, so it is totally possible to extract DNA and RNA from these lichens. I think that gives us a ton of valuable information. What we're trying to do in the lab is to get a sense of what's being expressed during some of those early points of association. And that's why it's a bit different from just extracting from a lichen that's already there. The lichens grow very slowly. I don't think I actually mentioned that. They don't just grow slowly in the lab, they grow relatively slowly in nature, especially when you compare them to other growing things. And again, that's one of those, lichens are polyextremophiles. Some lichens grow much faster than others, but it still ranges between like millimeters to centimeters of growth per year, so not a lot. But basically, by the time you're seeing a lichen, it is fully mature. And so it would be hard to, in nature, measure those early contact stages and what genes are being expressed. And that's the real, I think, push we're doing in the lab. I think the real original experiments were really just to see if we could create a lichen in the lab, but now it's much more focused on those early stages of association. Yeah.
I have two questions regarding the so for the establishment of the symbiosis relationship between the microbiome and the protobiome, I know there are a lot of people that know it, but there's some people already know it in terms of the molecular basis and also the morphological chain. What's the difference?
Yeah, so the question is, how does this initiation of the fungal-algal relationship in lichens compare to fungal pathogenic infections of fungal pathogens? In some ways, I want to say we're too early in the data to say for sure, but I think there are definitely some similarities, certainly in some of the signals used and in some of the other pathways. But I don't feel confident enough to say more than that right now. Yeah.
Another part is that the difference of kind of light, considering the field seems like the light can
me see if I understand your question. So lichens in the lab, the fungi will associate with the algae, and it seems to be in an exchange.
Yeah. There are other fungi that people know.
Yes, and can contaminate it. Yes, and so that is a mystery. That's one of the perplexing things, because yes, we know that lichens have all these secondary metabolites that help protect them against bacterial infections, other fungal infections, et cetera, and yet in like on growth medium, those other fungi can easily overtake and even destroy the microbiome that we're trying to culture. And so there may be something missing there, or it may be that they have to grow to a certain point in order to be able to defend themselves, or maybe they need their symbionts in order to have the full arsenal ready to go. It's a great question, and I think understanding the answer to that question will give us answers to a whole lot more things at the same time, for sure. I want to come to this side of the room. Yeah.
Over 10 years ago, there was a kindred species, Brazilian that rhizoids of this fungus, this zoosporic fungus, will capture an algal cell and live off of that same. You could separate the fungus from the alga, but the fungus would die. But the algae survived, similar to your situation. We had an impure culture, food culture essentially, just on agar plates. It's happy and content. We could freeze it, cryopreserve it, revitalize it. Is that a lichen?
Yes, I love it. By certain definitions, yes. There's some argument that basically this complex symbiotic phenotype has to arise from the association for us to recognize it as a lichen. So I guess one cell plus another cell is not quite enough to be a complex symbiotic phenotype.
On an agar plate, that's not like possibly a cauliflower kind of.
Yeah, so I would say maybe yes. My ecologist.
Really my question is, what's the fungal aspect of the lichen? Does it have to be an ascomycete or just pseudomycete? Can it be, if we had some sort of entomopteran that had an association with algae, is that a lichen? What?
Yes, no, I love this. I feel like, so it not only depends on the definition that you're using, it depends on the lichenologist that you ask. Because of course we're all very opinionated about what makes a lichen. I like to take the broader view, and I certainly don't think we should limit ourselves to ascomycetes and basidiomycetes, just because those are the ones that we have tended to study the most in terms of this association. If there are other things forming stable associations with the symbiotic phenotype, why not? I don't know. Why not? Yeah. And certainly, lichenologists already study what we tend to call lichen-like associations. So there's some really cool examples from the intertidal zone where macroalgae have fungi living within them. It's almost an inverse lichen, or is it just an endophytic fungus? But people have called it a lichen-like association, and so we study those too. So I think within that broader sense, totally, yeah. Somewhere in the middle, yeah.
So is there a push within the lichenology world to have a model lichen, like we have Rhabdopsis gray, to be able to push forward the...
I could be wrong, but I think that's happening in this exact department.
Okay.
Yes. It would be very useful, because really the next stage to get at understanding the nature of the lichen symbiosis is to start making mutants and either taking away or inserting genes and seeing what happens. Because I was talking to some folks, so for my postdoc I did this big co-culture experiment. We had all this RNA-seq data, but 95% of it comes back as NAs because we just don't have the genes characterized, and so it's very frustrating. So we can't get a lot of data from that. So it's time to start really characterizing individual things in light system. And I think that's happening.
But
So,
I guess
yeah.
my question is, maybe there's a lot of different labs trying to do that
Mhm.
with different species.
Oh, yeah.
And is there like some kind of consortium that is trying to
Ooh, that's a good question.
make this work great? Because it's really cool.
It's really cool. I know that multiple people are interested in it, but I don't know how many labs are actively pursuing it. I think there's been some cases made for lichen models, but more from an ecological standpoint. But now that we're taking it to this next level of actually trying to transform things, there might be totally different lichens that we want to be using for that. Yeah.
The ones that actually be able to resynthesize primitive lichen in the lab or fermenters and are difficult to do any genetics, molecular genetics. But the one we actually try to push is actually the omnidimorphic, the yeast facing. And interactology become hyper. So we actually could do molecular genetics, we can knock out genes. And then we try to propose this as a model. Of course, we want fun. But whether, at least we have two publications, but I don't know whether it will. Because the people actually traditional working on lichen resistance, the ones actually not amenable to molecular genetics.
Yes,
We just cannot do the gene knockout transformation.
Exactly. Yes.
And the system, the visiting scientists comes out, we actually put down yeast place, we basically purify the yeast transfer. Sorry.
No, I appreciate it. That's a great addition. And that's a really good point. That's why that is potentially a really great system as opposed to many of the other lichens we can try to work with. Yes.
The lichen that's too long.
Yes. So the question was, is there a phenomenon of lichen bleaching the same that we observe coral bleaching? It's kind of a yes and no. Absolutely, yes, we have observed this, but it never seems to happen at the scale that we've observed coral bleaching happening. So I do think it is a response that they have sometimes to various stressors, but probably a lot temperature. Sometimes a lichen, just like coral, sometimes a lichen that bleaches can recover and sometimes not. And I think there's a whole lot about that we don't fully understand because it seems to be sort of this patchy event as opposed to like a really dramatic broad scale event. And so it hasn't been investigated as much as coral bleaching, but I think it should, because I think maybe we'll start seeing more and more of it as the climate changes. I think we're out of time perhaps, but thank you all so much. Yes.
Oh my God.